I'm here to talk to you about the work I've been doing in Nigeria and in Bowie State as a teacher. These two institutions have a lot in common, Bowie State University and Godfrey Okoye University. So my overall goal, because of my history and where I came from, I actually came from that background in Nigeria, where I did my master's degree in genetics several years ago, before I came out to University of Maryland for my PhD. The background is that there's so much teaching going on in those institutions, so much theoretical knowledge. The students graduate from those schools knowing everything that you know, except that they have never done what you have done. Many of them have not handled pipettes, and they have not seen primers, worked with them, but they do have the knowledge. So I began to think, I became very determined to do what I call targeted infusion, which is to identify those colleges where these things are happening and try to, where they have the programs, biotechnology programs, and try to bring in laboratory experience for the students. And DNA Subway, offers itself for this kind of experience. I met the DNA Subway, the iPlant team some years ago in the conference of this American Society of Plant Biologists. And since then, they have come to my school. Actually, that's another thing you want to know, that they can actually come out to your school and do a workshop if you invite them. So after they came to my school about 4 years ago, we became friends, and that's how I got to be doing some of the things I'm doing now. So DNA Subway provides an educational platform for gene annotation, phylogenetics, and transcription. And I'm not going to be repeating this because a lot have been said. You have heard about the lines. And I'm going to be concentrating on the blue line. The blue line enables easy analysis of DNA barcode data. And the projects that we do, the type of projects we do actually exposes our students to so many things, to different kinds of things that they will not normally get in one class. Because usually you teach a bioinformatics class, you teach an evolution class, you take a biochemistry class. But in this one module, you are able to expose students to everything at the same time in a course of one semester. Our DNA barcode sequences are automatically uploaded into the system. And a lot of, like Jason said, a lot of softwares are built into it that enables you to get the work done. So what has happened in recent times with me? Before this time, I used to teach. And the way I taught was, I thought about skills, equipping students with skills. The skill for DNA extraction, the skill for DNA fingerprinting, the skill, in a very disjointed format, but not with a research question. There's no, usually in the classroom, okay, I go to my lab and I do research with students, but in the classroom and labs, we don't look at a research question, no inquiry being made about anything. So in the past 2 years, that has changed, because now my labs we build in research into it, and the students understand the question. It's open-ended, and they can come out in any form they want based on what the discovery that they are making. So students are able to use the blue line to analyze data and publish sequence results directly to the GenBank within the DNA Subway itself. They don't have to get out of it. Right there, they can complete the work and submit their sequences to the GenBank. So to implement this in Godfrey Okoye University, our aim really was we started a project called DNA Barcode Initiative for Species of Eastern Nigeria. And again, that was exciting to me, because now the people in Enugu State are benefiting from this. Why? Because there are so many species that are in that part of the world that nobody has looked at, nobody has investigated. So our specific objectives were to have students identify unknown species using DNA barcodes, to discover new polymorphisms within existing ones and establish phylogenetic relationships, and, of course, introduce the new species to the GenBank. The barcode production pipeline will include sample collection and photography, extraction of DNA, amplification of DNA using PCR, electrophoresis to check PCR product, and sequencing of the amplified data segment, then followed by data analysis. Now, embedded in all this are all the techniques that you really would like your students to have. And we are talking about people who never pipetted anything before, mostly, from where I'm coming. They have not done pipetting, they don't understand, they haven't done DNA extraction before. They don't understand PCR and how to do it. They haven't even seen PCR machine. We had to provide that for them the very first time. So they are learning, they are making inquiries, and they're acquiring those skills. One of them, Mike, became very outstanding because in a short time he mastered the techniques and began to work on very expanded projects. And Mike did so well that he has already he's presenting his data at international conferences, and he's being considered now for admission into the PhD program at Watson Graduate School at Cold Spring Herbal Laboratory. So he may be coming out here this August to start his graduate work. Again, am I excited about this? I am. Why? Because it could not have happened if not for this program that we start this collaboration with Cold Spring Herbal Laboratory and the DNA subway and what it permits us to do. So the summary of outcome: research culture and undergraduate research is enhanced in both universities. The undergraduate research experience appears to improve students' learning and biological expertise, open more doors for postgraduate work in molecular biology and biotechnology in both universities.